loops. So there are different criteria by different researchers, and we combine them to all of those filtering criteria to remove the false positive, because when you go to the computational approaches, the chance of the false positive is higher than the wet lab. And then finally, when to apply to remove the false positive, finally we got the new conserved microRNAs. And in the wet lab approach, small RNA library approach, first take the total RNA from the tissue, from the cells, from the plant, from the animal in which you are interested. Second, isolate the small RNA with the help of different kits. One of them is like Mega microRNA isolation kit, and next, small RNA purified through this kit will be enriched through polyacrylamide gel electrophoresis page for 18 to 13 nucleotide molecules, and then ligate the adapter at the 3' and 5', and then go to sequence it with the next generation sequencing, and then next go for the bioinformatic analysis to develop the secondary structure and other things. So this is the wet lab of the small RNA library. And these are the different our lab reported microRNAs, the organisms, and we reported first in cotton into the 22 number of microRNA was 22, it was 2008. During my PhD, my work is on the cotton, and I am working on the microRNA on the studies of the vaccine, not on the microRNA. I am working on the microarray. Now you are familiar that microarray is the quantification expressional studies of the messenger RNA, and also the differential gene expressional studies of the messenger RNA. So at that time, I have one question: is there any chance that a messenger RNA form but not translate into protein? Because the protein are the working horses. We use the term working horses. They are the functional unit. The messenger RNA is just an intermediate molecule in the central dogma of the molecular biology. It is not the final one. It is not at the end to perform the function. The protein are the final to perform the function. So there was a question in my mind at that time: is there any chance that messenger RNA is present but protein is not synthesized? And at that time, when I go to search the literature, the other thing, then I found that the microRNA are the molecules which can do this with the messenger RNA. That messenger RNA is formed but not translate into protein. So it means the microarray are the messenger RNA level studies, although it is good but not fully helpful to got the answer that they are going to be translated or not. So then I will focus on the miRNA, and I will form 22 novel miRNA in cotton, and we reported them in 2008, and then between I focus on my PhD, do my PhD, and then work on the mimulus and report 34 million species, 61 sheep, 172 salmonella, 102 China crayfish, 73 flax, 26 Artemisia 16, Bacillus 55, carrot 17, tomato 109, and eggplant 76. And these are the some results that I found here at TSU in the switchgrass through the dry lab or the bioinformatic approaches. So few of here. Well, till now I'm still going for the manual inspection to remove the false positive. But at this stage, I have identified 100 plus. But when we go for the manual inspection, it will be reduced to 80 to 90. But still, the entire database is not, the entire reported microRNA are not completed. It will go further and it will reach to 100 plus. These are the few that I found here in TSU in the switchgrass, and the green sequences representing the mature one, and they are present in the stem region of the stem loop sequences. These are the precursor microRNA structures. And I also found the cluster, one of the cluster, the microRNA, one mature microRNA is present twice in the one precursor microRNA, and it is PBI-MIR2275B. So you can observe here are the two greens. So one mature is here and second is here. Both mature sequences are same. And then I also found the sense and anti-sense cluster. That the sequence contained the two microRNA in sense orientation, and it also contained the same two sequences in anti-sense orientation. That is the reverse order of the first one, is the anti-sense or anti-sense. So that means this is very interesting that in the switchgrass, I found almost all the types except one, which maybe I found later, that is the overlapping microRNA. But the remaining all are found here, the sense, anti-sense, the cluster, and the 5', 3', all of them. And I acknowledge at the end the Fulbright administration providing funds for my visit here. I really appreciate Professor Zulsopin for providing the opportunity to come here, for accepting me as a scholar here, and I also acknowledge Dr. Aziz Ahmad, who helped me to settle here in Nashville, the beautiful city Nashville. I also thank to Sami Kumar, who helped me in correspondence and initial settling. And I also acknowledge my lab member here in TSU, the Juja, the Shashi, and all, and also at the University of Balochistan. So the house is open for questions. Welcome to your questions. The questions are guaranteed. The answers are not. Yes.
Okay, can I ask, the numbers don't seem to go very high, except recently in the switchgrass we see 2,561 for the coding of the sequential discovery. So when you do a wet lab experiment and you discover so many, do you immediately put the numbers and define, or do you have to wait for a long process of determining conservation and orthologous and relationships like this? For example, because I know in beans, there's several groups in Mexico doing the microRNA, and they've sequence, and you told me a surprise now that it's not in gene bank. I thought it was in gene bank. So I'm curious about the numbering and the no gene bank. Would they require a gene bank?
Actually, what I got from your question, I'm going to answer in that way. If you identify the conserved one, already reported in some other species, in some other organisms, then you not need to give the other number to it. Just only change the first 3 letters for the species. Like here we did, like we changed just the, this is the we found it through the rice. So we remove the rice OSA with the PBI, the number remains same, 2275 is the same. When it is conserved, already present in the others, the number will remain same. Only the organism name, 3 letters will be replaced. So, and when it is new, then new will be numbered according to the previously order. If previously the last one is the 6002 and someone discovered a new one, it will be named 6003. But if it is some sort of the ST sequence, some sort of the GSS, some sort of the RNA sequence or DNA, it should be submitted in the GenBank. But for the microRNA, it is not essential. Although small RNA libraries are submitted to the GenBank, but microRNA it is not essential to go to submit the microRNA sequences first to got the accession number, and then you will be able to publish it. What I found in, during my experience. It is not essential, as in the case of the EST and others.
And species specific?
Yeah, there are some, yeah.
Yeah.
If you have, also for the species specific.
Will they tend to be those higher numbers?
Yeah. To use the higher numbers, to use the next number for them. But sometimes they again start the number 1, 2, 3 in their paper that we found the new one, and then they call them the new potential microRNA 1, new potential microRNA 2.
Not 25,000, because in all the species there were 25,000, so how do you...
Yeah. But sometimes, yeah, both approaches were used by the researcher. Either to start the new numbering for their work, or to continue the old numbering as they found in the literature.
I'm not sure if you talked about it before I entered. I entered halfway. But what is the difference between mature and precursor mRNA?
Precursor is a stem loop. There are many different, but you have to remember two basic differences. First, precursor length is more than the mature. The precursor length in animal is 70 to 90 nucleotide, but in plant it is up to 300 plus nucleotide reported. And the mature length is small, it is 18 to 26 nucleotide. In the average or the mostly observed length for the mature in plant is 21, and in animal it is 22. But the range is 21 to 26. And second, the precursor, due to self-complementary, it developed a stem loop structure. And in the stem region, it contains the, like here, this is the precursor sequence, started from 5' ending at 3' single strand, but due to having complementary sequence, it folds with itself to develop this structure. This is called stem, and this is the loop, and the stem contains this mature one, which highlighted here by green color. So it means the mature is a part of the precursor.
So how do you know, like, what part is the mature? I mean, how do you know if you identify a new? Because if it's conserved, you probably know. But
yeah, I have already given the idea. Maybe you not. Okay, I'm going to explain it. No problem. If you're going to on the basis of conserved, no problem. You take one already reported, confirmed as a mature. Find it similar here in other species, so it is the same. For the new one, you go to isolate the total RNA, and then from the total RNA, small RNA, from the 18 to 26 nucleotide length, go to sequence them, got the sequence, go to the bioinformatic analysis to find is there folding, is there the secondary structure formation, are they going to be present in the stem region, are they be the part of the non-coding RNA, are they be the part of the some sort of the 3' untranslated region of the mRNA, and they then confirm that these are the microRNA.
Okay, I think I have a suggestion. Based on Dr. Barozai's presentation, I think we need to have a whole afternoon here.
yeah.
And I want everybody to be here if you are interested in. Bring your own computer. We'll set up a session for afternoon. Can you do that, Dr. Barozai? Yeah. So we get online. Everybody connect your own computer. He will upload it. Have a presentation on the procedures. So like hands-on practice. So will that be a better idea?
That's a good idea.
And then let's do it the next week. Before I go, I want to know. I want to learn this. So I'm going to send everybody an email. We'll do this next week. We'll try if we can do it in this room. Because I know the CIT is fully booked. We should know, we should have got this done. We'll try it this room. Everybody bring your own computer, it should work. So based on what's so much information in it, I think we plan start at like 1 o'clock, go through the whole afternoon. So if we still don't have enough, we want to have one more session, Dr. Barozai will be here until the end of this year. So anybody who wants to learn, just send your suggestions. We need one more session, and then we'll have one more session. We want everybody here take advantage of this opportunity to learn this. Okay. Because only by explaining, this, that, you probably just, you may understand now when you walk out this room, you forget completely. So we give everybody 1 week to go home, look at the art, look it up, or maybe you find some examples of microRNA from the species that you are working on. This is regardless of your animals, plants, it doesn't matter. And put together your questions you want to ask. So we will have one afternoon next week. We'll do that. Yes. Anybody have more questions?
One later.
Yeah.
One or later. So congratulations, very clear, and this is a new area, so putting it all together and then giving the examples to scratch is very beautiful. I have a question. When you look at a genome, for example, if we were to bring a genome
Yes.
on our computer, maybe is that too complex? When you look at an entire genome, you said the dry lab approach, do you find these basically inverted repeats? There used to be a class called mice. They were called micro-inverted transposable elements, or something like that. And they were transposable elements that moved around, and they contained a simple sequence repeat that then had a potential for hairpin if they were expressed. Is there any relationship that you can do this kind of searching in a large genomic context? For example, if I brought 100 kb of now let's say even more, let's say 500 kb of information from a genome, would I be able to find probably a microRNA?
Maybe some one or two or many microRNA present here. So we can use them as a source to blast them against the reported one and to find either it is DNA or RNA, any sequence.
Okay. And potentially, if there's 25,000 existing now, would you say that every plant would have how many number of these kinds of loci? Because they're not really defined as genes in the old,
till now, the famous Arabidopsis. In famous Arabidopsis, there are 1,000 plus reported. So from there we can take an idea that up to maximum 2,000 to 3,000 microRNA per organism or per species, in which mostly are conserved and some are species specific. But they are conserved. There are differences in the precursor, in the mature, in the mode of action, in the mode of targets. And one other thing, when you use the term transposons, so the microRNA also help to stop the transposons from their action, from their jumping into the genome where they cause problem for the organisms. And there is also another interesting class of small RNA which is siRNA, small interference RNA. Those are also endogenous, small in size, same mode of action as microRNA, but not going to form the stem loop structure. They are also abundant, and they sharing 50 to 60% similar approach as by the microRNA. They develop a duplex the duplex contain one specific siRNA, and it go to the risk RNA-induced silencing complex, and it go to the messenger RNA to silence it. And there are also new type of small RNA which are reported nowadays, and means this the small RNA world in the plants or in the animal is very big, and they are doing the regulatory role. And nowadays there was a publication which reported that the plant microRNA, when taking the juice of the plant by eating the plants, it go to within the animal or within the human, and it also play a role there. Although still it is debatable, still not as famous as approach, as good accepted approach as the other, because microRNA, if it goes for the digestion, it will degrade up to the nucleic acid, ribonucleic acids, uracil, and the adenine, and the cytosine, guanidine, and then it will be able to absorb in the blood. But still they saying that there is some protection mechanism for them for this 18 to 26 nucleotide strand, and this protection mechanism is provided by the protein and lipids, and it also help to protect it from the digestion and also help it to ingest in the blood. And when it go absorb in the blood, it their work for the organisms. So this is the reason that mostly in juices or in fruits or in vegetables that we taking, and we saying that these are the good for the human. So microRNA are also in them, and they also doing some job in there. But still not famous, still not, because if some sort of the macromolecule we take in the ingestion, it will be digested by the enzymes up to the monomer level. So, at the same time, I will want to share the one of the journal that we started, research journal. Submit a review, particularly the student, to review a research article to one of the journal where I am the chief editor, and I will, as attending this seminar, this presentation, I will give you the favor to not charge the publication charges. So in your field, any review paper or research paper, if you go to submit it, the journal name is Pure and Applied Biology. You write it. Pure and Applied Biology, and its URL is the Pure and Applied Biology. And its site is the dpape.org.
I'm sorry.
dpape.org, particularly for the student to initiate.
Are you going to have a special issue?
pure and applied biology. Covering all the sides of the biology, and it is a new journal, just 2 years it has been done for publication. We had some indexing in some, like the Global Impact Factor, Google Scholar, Visra, Europe, ProQuest, GeoImages, CAS, SciPicture, Agricola, and others, but still not the ISI impact factor. This is some other institute impact factor, not the ISI. So I want to clear this. It is not the ISI. It is another institute from the Australia. They are providing us the impact factor. So, and mostly I recommend for the students, initially they learn to publish a paper. So a review paper or some sort of basic data you have. So you try to serve, because we charging $100 US, but for you, as a reward to attend this seminar, we will not charge. Okay.
Wow. You got recording already. Schedule and topic to the TI, and then they will distribute to your own members. Is that okay? That's right. Yeah. So I think that today is Tuesday. That's Monday. Next, what's the day of next?
17. 12.
Monday is 19, so we are leaving on Monday.
Tuesday.
We are leaving on Tuesday, so let's plan on next Monday. Will that be okay? EPA has to be okay, so next Monday. Yeah, Monday is
So
good.
you come back. You come back. Sunday.
So, tentatively, let's plan on that. Will next Monday work? Yeah, so next Monday afternoon, 1 o'clock. So we'll meet in the same room. Yeah, before that, come bring your own computer, make sure you need to know. If there's any changes, we'll let everybody know. Yeah, I think it's very, very interesting. It's something we want to start in our research, trying to. Actually, Dr. Barozai and Julia, and he mentioned your RNA may be transcribed into mRNA, your DNA, and the mRNA may not be expressed into protein. So what they are doing, they are trying to do one experiment, take one set of samples, and we are going to analyze microRNA, mRNA, and protein. Everything is the same from the same tissue. So we can find more dynamic changes. Okay, thanks for everybody.